So hi, everybody. I am thrilled to have Laura Shawver, PhD scientist and ovarian cancer survivor, who's going to discuss with us treatment landscape around clinical trials and how to be more informed on what clinical trials might be best for your individual journey. The Clearity Foundation helps finding a clinical trial, knowing that it's daunting because there are just so many. Laura will walk through how to use Clinical Trial Finder on their website, which is a link that was sent to you guys, to quickly hone in on specific trials more tailored for you. She will also be available to address questions on tumor molecular profiling and how to obtain these services if desired. So I'm going to turn the floor over to Laura, and thank you so much for joining us.
Great. Yeah, thanks for the opportunity to talk, and nice to meet every one of you. I know we don't know each other, but I feel like we have something in common, and I know that this program is very, very helpful for all women who are have had a cancer diagnosis. We're going to be focusing on ovarian cancer today, but of course the principles do apply to any gynecological malignancy or actually any type of cancer, to be honest, although our clinical trial finder is specific for ovarian cancer. So what I'm going to do today is I'm going to share 2 introductory slides, and then I'll present an agenda and then walk through the agenda. I only have about 8 or 9 slides, and we're going to, in the middle of it, watch a 7-minute YouTube video. And then we're going to do live the clinical trial finder. Maybe one of you will want to share your information, but if not, I can do my own, or I can do somebody else that I've recently spoken to. Okay, so just be thinking about if you want to share any of your information. So you may be familiar with Clearity Foundation, but perhaps not. And we are trying to help people one person at a time, because we do feel that everybody's experience is unique. And we have to be treated in a unique way. And I'm going to talk more about that, and particularly about my own background. But we are, as an organization, trying to help each person understand their diagnosis, their treatment, their life situation. And we're not going to talk today about the Steps Through program. I'll mention it at the very end. But overall, we really want to give women with ovarian cancer more options and more hope for their individual journey with ovarian cancer. So altogether, Clearity Foundation does provide a number of services. We're a not-for-profit organization, and all of our services are provided without any cost. So we do have professional one-on-one support. And at another time, if you want to hear about the Steps Through program, we're happy to have a conversation about that. I'm probably not the right person, but Susan Hess, who heads that part of the organization up, she would be delighted to have a conversation about this. We have a ton of educational content, for which we're going to talk about today, as well as, the resources, the expert resources, of which I am one of those, for helping to find a clinical trial and talk about this whole concept of molecular profiling or tumor profiling, and really trying to understand what makes our tumors, grow and divide. We know a lot of gynecological oncologists out there, and we oftentimes provide referrals, particularly for, those sites that have clinical trials running that might be right for you, but aren't running at your oncologist clinic or organization. And we also provide group support. So that's the two introductory slides. And what I thought I would do is to talk a little bit about my background and why I came to found Clearity Foundation 14 years ago. It's hard to believe, actually, that Jillian, I think there's somebody else in the waiting room. It's hard to believe, actually, that that much time has gone by, but we've had really tremendous learning since then, and I wanted to touch on that. And then, of course, the main focus of this is the clinical trial finder and how we can help should you want to consider a clinical trial. So I'm just going to pause here to see if there's any questions before I go on. Okay, so a little bit about me. So I'm a scientist. I'm a PhD scientist. So I'm not an MD. I'm not a medical doctor, and I don't give medical advice. But I have been working in oncology drug development since 1989. And actually, I got my PhD in the early 80s. And I'm trained as a pharmacologist, which is the study of drugs in the body. And I was trained as a pretty classical pharmacologist, meaning not just the study of drugs, but how they're metabolized and the toxicology or adverse events that come up from drugs. But in the early 80s, it was really the dawn of cloning and how do we clone genes. And I wanted to learn how to do this. And of course, back then it would take a year to 2 years to clone one gene. And now it's amazing because actually any high school student can do it in a weekend. And that's how far the field has come. But back in the mid 80s, I wanted to learn how to clone, and I retrained myself as a cell and molecular biologist through 2 postdoctoral fellowships, one at University of Virginia Cancer Center and one at Washington University in St. Louis. And then I serendipitously landed in biotech in 1989 because I answered an ad for somebody. Back then we didn't have any online tools, and you had to look in the back of journals to see who had jobs. And I answered an ad for somebody who had with experience in oncogenes, growth factors, and signal transduction pathways. And so oncogenes is this, genes that cause cancer, and how things signal from outside of the cell to the nucleus to cause a cell to grow and divide. And I thought, oh, that fit my background perfectly. And that's how I landed in biotech at a company called Triton Biosciences in the Bay Area in California. And I started as a bench scientist in the molecular biology department. So that's going to come back in another couple of slides about what I was working on back then. But since then, I've been employed at a number of biotech companies, all of which were working on understanding cancer treatment based on molecular alterations. So as you know, when people say, oh, I have cancer, they say, oh, I have lung cancer, or I have breast cancer, or I have ovarian cancer. But that's not how we define cancer as scientists. We define cancer by what's the molecular alteration. Like I have HER2 amplified cancer, or I have EGF receptor mutated cancer. So anyway, I did in 1989, as I started in biotech, it was the dawn of precision medicine. So how do we match the tumor and what's wrong in the tumor with a particular drug? Okay, so in 2006, I was out, I live in San Diego, and I was out surfing one morning and I felt a very weird pressure my lower abdomen when laying on my board. And I thought to myself, I better go in for my annual pap and pelvic, because this was in August. I thought as soon as September hits, I'm going to be busy again. I'm not going to have any time. Well, long story short, that ended up with an ultrasound, and that ended up with a diagnosis of ovarian cancer. And when I had my surgery, I learned it was ovarian clear cell carcinoma. Some of you may know that that's a rarer form of ovarian cancer. Most people have high-grade papillary serous, but I had this rare clear cell carcinoma. So I had the surgery, same as probably most of you, and I had 6 cycles of carbotaxol, probably the same as most of you. And when I, was meeting with my surgeon, I told him that I wanted to have my tumor profiled. And he didn't know what that was. But back in one of my companies, and I'll come back to this in a minute, when I was working in the lab and we were running clinical trials, and we're going to talk about clinical trials today, we would get samples of people's tumors that we were doing clinical trials on, and we were studying in the lab, and we were trying to understand, what their specific alterations were. So that's what I wanted to do. And I even made one of the people in the lab in the company that I was working at the time, I made them go and get a piece of my tumor from the operating room and freeze it in liquid nitrogen. And I stored it, in a -80-degree freezer for a long time, wanting to get my own tumor profiled. Eventually I did. I'm not going to talk about that today, but I did at the time find out that Dr. Laura Shawver, the scientist, could profile other people's tumors in the lab, but I couldn't get my own tumor profiled to save my soul. Okay, so that's a little bit about my background. And now, as time has gone by, I have come to lead companies, and I'm currently CEO of a company in San Diego called Capstan Therapeutics. Okay, so now we're going to have a pop quiz. I thought this might be kind of fun. So here's a question for you. What was the first cancer drug approved based on a molecular alteration, and what year was it approved? So I'm going to pause to see if anybody has an answer. Does anybody want to guess? Okay.
Well, I could guess
Keytruda.
that.
Keytruda.
Yeah. Pardon?
Keytruda.
Okay, that's a good guess. Anybody, any other guesses?
I'm not sure about which drug was approved, but I think it had to do with breast cancer.
Bingo. Who said that? Okay, well, awesome. That's exactly right. So
Because
it was
that when the breast cancer tumor was the first oncotype that was studied.
exactly right. That's exactly right. So it was Herceptin that was approved in 1998 for breast cancers that had a HER2 amplification. So if we go back to 1989, when I started in the industry, the first paper was published on how amplification of HER2 meant a worse prognosis for women with breast cancer. And some of you may have friends that had breast cancer, which back then, if you had a HER2 amplification, there was nothing that could be done. It was a horrible prognosis, and people typically passed away within 6 months of the diagnosis because it was so aggressive.
Can I ask a question?
Yes.
With the HER2 tumor, that's different than triple negative. A triple negative would not have been HER2 positive, right?
That's correct. In breast cancer, it's divided into 3 major subtypes: ER positive, HER2 positive, and something called triple negative, which does not express either ER or PR or HER2. So this
But I
is,
always thought that the triple negative was the worst of the three prognoses for breast cancer.
it is now, but it wasn't back then.
Well,
So,
that's what I understood back then anyway, but I'm
yes,
glad you're clarifying it.
yeah. So now, it used to be that if you had a HER2 amplification, that was the worst. Now we have a treatment for it, and it no longer is. In fact, women live a long, long time, and hence triple negative is the problematic subset now. Although there's a number of treatments now for triple negative as well. I don't have time to talk more about that today. So here's a cartoon of what this HER2 is. It's on the outside of the cell, and when it signals into the nucleus for the cell to grow and divide. And Herceptin is a monoclonal antibody that blocks that cell signaling. And so again, back in my first job, one of the things that I was working on was a HER2 antibody, and how to modify that antibody to take it into clinical trials. Now, that particular company did not take its HER2 antibody into clinical trials. It was Genentech who did that, and they were the ones that got this first approval in 1998 for a HER2 amplification. So do you guys want to see what things look like today? Here we are, 30 years later, 98, 2008, 25 years later. Do you want to see how things look like now? And it took so long to get one drug approved. Here's what it looks like today. So if you look down here at the HER2 or B2 gene amplification, trastuzumab is Herceptin. So many of you probably don't know this, but drugs have two names. They have a generic name. The generic name is listed here on this slide, and they have a trade name. And so trastuzumab is the generic name and Herceptin is the trade name. But it does target these HER2 gene amplifications. And so you can imagine back in 2006, when I was diagnosed, we knew about Herceptin, we knew about EGF receptor alterations, we knew about ALK translocations, we knew about these PI3 kinase alterations. Where are they? Here, PI3 kinase alterations. And there were not as many drugs as there are today, but there were a number of drugs that were approved. And I thought, oh, I should get my tumor profiled because clear cell carcinoma is very rare, and I might have one of these molecular alterations that would match with one of these drugs. That's why I founded Clearity Foundation, to help other women get access to get access to be able to have their tumors profiled and understand the results. So whoever mentioned Keytruda, it's right down here. It binds. It's called a checkpoint inhibitor. There's a number of them. There's like 6 of them, so they're not listed here. But Keytruda is one of these. Okay, so you see that there's been a lot of progress made, and it can all be so confusing. Because if you think about, it takes a PhD scientist who's grown up in this environment, and I've watched, how we've understood the various alterations that can make a tumor cell grow and divide, and watched as new drugs have been developed over time. You can imagine a person not familiar with this, just how confusing it can be. And then there's all these other drug classes that we're not talking about here, but are just as important, particularly as it relates to our treatment for ovarian cancer. chemotherapy, for example, angiogenesis inhibitors, anti-hormone therapy, and then important when to consider clinical trials. And in addition to considering each of these, the precision medicine drugs, chemotherapy, angiogenesis inhibitors, anti-hormone therapy, there's new classes too, these antibody drug conjugates, and then immunotherapy. Keytruda was mentioned, that's one of these immunotherapy drugs. So if we as somebody diagnosed with ovarian cancer or recurrent ovarian cancer, we're considering a clinical trial, if our oncologist is considering a clinical trial, it can be very hard to know which ones are right for us. And last night I did a quick search on clinicaltrials.gov just for ovarian cancer studies that we're enrolling right now, not all the ones that have been done in the past, but just today, 382. So how do we sort through all of those? Okay, so should I get my tumor profile for molecular alterations? Well, we launched in 2008 with that as the mission to help women with recurrent ovarian cancer get access to the latest molecular testing. Oh, and it was hard back then. We had to fight for every person who contacted us to get their tumor profiled. And I'm not going to go into all the details about how we did that, but a lot has changed since then. And now it's in the NCCN guidelines that everyone with recurrent ovarian cancer should have their tumor tested for molecular alterations. And I'm going to show you that right here, because it's important, because that the fact that it's in the NCCN guidelines means that your oncologist can prescribe it and your insurance will reimburse it. That was the other thing that Clearity did in the early days, is that we believe so strongly in this that if somebody wanted their tumor profiled, but they couldn't get it reimbursed by insurance, we actually would pay for it. So you see here, this is the NCCN guidelines. I put the link below. It's a complicated document, but you see anybody with ovarian cancer, with recurrent disease, tumor molecular testing if not previously done, tumor molecular testing if not previously done. So this was a big change from 2008, and for the first 10 years of the organization, how do we get this for everyone? And so today we can direct you to testing facilities if needed, and we can discuss the report with you, because these reports can be a little bit daunting. And we have people other than myself who can help understand that and help guide the choice and explain why it can guide the choice of a clinical trial.
Can I ask a question?
Yes.
Is the tumor molecular testing the same as DNA testing?
It can be, yes. So it's called different things. It's called molecular testing. It's called genetic testing, but genetic testing is really divided into two categories, because you can take a tube of blood and be tested for germline mutations that we're born with. And most of you may know that BRCA mutations, something that corresponds to PARP inhibitors, are causative for ovarian and breast cancer. And that's something that's that women are born with. About 10% of breast and ovarian cancers have a germline BRCA mutation. So genetic testing oftentimes refers to testing from the blood versus tumor testing, where you actually need a piece of the tumor, and that piece of the tumor is sent to the lab. And that they do isolate the DNA from our tumors and look to see what the alterations are in the DNA. It's a good question.
Were there any other questions before Laura continues?
I have one.
Sure.
I actually started to type it, but it's probably faster if I say it. So I had my tumor tissue tested after my second surgery in 2016. But,
Okay.
but now I'm still at it. It's 2022, and you know I'm actually living with a recurrence at the moment. They tried to biopsy a new spot last year, but it was unsuccessful. So I guess my question is sort of two parts. I mean, one is I paid to have tumor tissue from that 2016 surgery stored, as you do, you know, cryogenically. I have frozen fresh tissue, but as I've been in it for so long, isn't it possible that there have been changes that now that you know that what's been saved from 2016 isn't really the same as what exists in my body now?
That's a great question, Mimi. So first of all, for everybody listening, when we have our surgeries or for primary removal or for recurrence, the path lab stores formalin-fixed paraffin-embedded tissue for 10 years. So your path lab that you had your surgery back in 2016 still has these FFPE blocks, and most of the tumor molecular profiling is done on these FFPE blocks. We no longer need frozen tissue. And I would encourage anybody that is in your situation who maybe had a profile done back a number of years ago to have it done again on the stored tissue, because there's been new tests introduced and where one might glean additional information. And if what we know about ovarian cancer is, at least on the molecular level, for certain types of ovarian cancer, it doesn't seem to change that much. But if we do have a new biopsy, yes, absolutely, get it done on the most recent sample that you can. And do you know where you had the testing done?
Foundation One did the initial report, and I actually, because I was in a clinical trial, so they ran it on the stored tissue, and
Yes.
my oncologist at Sinai ran the report through Caris. So I have
see.
two different reports from that 2016 surgery. Plus, like I said, I'm also paying. Well, it's up to now. It was $49.95 a month. Now it's $69.95 a month to store that the fresh tissue that was taken from the surgery that went to this company called Store My Tumor. So if I understand what you're saying, I really don't need to be paying for that anymore. There's no point in that they would be able to do anything with that tissue.
Well, first of all, you know, I think everybody needs to make their individual choice about if they want to store their frozen tumor. And people do it because they think there might be advances in the future.
Yes.
But most of the advances are being made on formalin-fixed paraffin-embedded tumors. And if you could please reach out to Jillian, and she will give you my email address, and I'm happy to look at your Foundation One report and your Caris report and have a conversation with you about it.
Well,
Okay?
actually, you know, I've been at it for a while with you, with your group, and with Deb.
Oh.
You've got my stuff. It has been.
Oh, it's okay. I can go get it from Deb.
It's more towards, and I don't want to steal from everybody's else's chance, but there's this one spot that isn't responding right now to the drugs that I'm on. So
I
I've
see.
actually, but that was the spot that they tried to biopsy, but they couldn't get any. It was an unsuccessful biopsy, or I should say it came back benign, which tells me that they missed getting what they needed to get. But So now I've agreed to go for radiation to have it eliminated, because that seems the less invasive, smarter path. And if Mary would want to put in her 2 cents, she actually listened to something, a webinar yesterday that talked about how sometimes certain spots are not related to the existing disease.
Yeah.
But how do you figure that out when there's only a small sample?
Yeah. So let's come back to that at the end. I want to make sure we get to this tutorial on the clinical trial finder. And then I'll come back to, how to think through spots that can't get resolved where they can't be biopsied. Okay.
Okay. Thank you.
Yeah. No worries. So thanks that was a good discussion.
I have a report, but need a deeper understanding of it. So is that something your organization can help with? Because these medical terms are not as familiar to me, but I'd like to understand my situation as much as possible, because I think it would empower me and let me know where I stand.
Yes, that's what we do.
Great.
We can help you with that and have a phone call with you and a Zoom call with you. And either myself or Dr. Annette Buell will do that. Okay. Okay, very good. Thank you. Okay, so we're going to go on now to talk about our clinical trial finder that's on our website. And when you go onto our website, there's this section on clinical trials that's this would be the landing page for Find Clinical Trials. And you can see that there's different buttons here. And I think that the best thing to do now is to just watch this 7-minute tutorial. I'm going to see if this pops.
Hi, my name is Deb Zykowski. I'm the Scientific Director of the Clearity Foundation. This is a tutorial for our clinical trial finder, which will help you identify clinical trials that best match your situation from amongst hundreds of trials available for ovarian cancer patients. We will start by entering information about your clinical situation, which can be newly diagnosed, in remission, or cancer that has not gone away or has come back. For the example that we're going to show today, we will choose for a recurrent setting. So we'll click on the button for my cancer has not gone away or has come back. Further specification of the clinical situation is next asked for, and the button to click in this case would be just recurred. This takes us to another screen with 3 additional buttons on it, in which we ask for information about cancer and treatments, genetic testing like BRCA status and tumor testing results, as well as information about preferences, such as location of the trial, phase of development, or the actual drug name or type of drug that you're looking for. We'll start by looking at the information for cancer and treatments. Keep in mind that there are many questions asked under each of these buttons, and you do not have to enter any information here. You can do the search without doing so. The more information that you enter, the better the trial matches will be. It will be more likely to be those for which you are eligible. So each question has a pull-down menu that gives you options to select. It also has a question icon that provides further information. And if you want to close these pop-ups or the pull-down menus, all you need to do is click outside of any of these boxes. So for the first example, we're talking about the histology of your cancer, and that is found on your pathology report. You can click the best match for your histology here. I'm going to choose high-grade serous as this example. Then we ask the drugs that you received in your first treatment, Most women receive carboplatin and Taxol, so I'm going to click those. The drugs that you received after your cancer came back or progressed. And in this case, we're going to assume that carboplatin and Gemzar was being given. That means that this example patient had 2 prior treatment regimens, the first one and the second one at recurrence. The next question is a really important one. It has to do with the number of chemotherapy regimens that you've had. More information, again, is found with the question icon. So in this case, I'm going to enter 2 prior treatment regimens. And the other question that's particularly important to answer, if you know the answer, is how long it was before your cancer came back after the last time you had platinum. In this case, I will enter that it recurred 6 to 12 months later. You can enter information about whether there's disease found on a scan and other information as well. The information about genetic testing and tumor testing is also given as an option for you to fill in. If you have a BRCA test that was positive, you would fill that out here. If it was negative, you would say none. And then information about the tumor is also available for you to enter if you have a tumor profiling report done. Now we're going to move on to the next and last section, which is the section about your preferences regarding the types of trials that you want to see. Are these trials only in phase 1, phase 2, or phase 3? If that's what you want, you would click. You will, of course, get the most results back if you leave that empty. The locations in which the trial is enrolling can be identified here. I'll choose California and Connecticut for this example. Obviously, you can choose other states. And if you do care about the kind of therapy that is offered in the trial, you can select a drug category. Some women want trials that have PARP inhibitors in them, so they would click this category. Others want immunotherapies. So in that case, you might want to click all of these categories. And then we'll do the search by pushing this button. This search will, of course, retrieve all of the trials for which you have a high likelihood to be eligible. And it's a long list, and it's ordered from the latest phase down to the earliest phase. The title of the trial is given. The locations in which it is enrolling is provided. And the eligible participant is also listed in a very shorthand way, as well as the phase. You can learn the drugs that are in the trial by opening this button, how those drugs work. And importantly, you can go directly to clinicaltrials.gov to see a larger description of this trial. It's important to look at the eligibility criteria at the bottom of any trial that you select, just to make sure that indeed you think you're eligible for the trial, because there may be questions that we have not asked or that you have not answered that will be critical for your eligibility. And finally, you can also save these results by clicking on the Save button, which will bring up the location on your computer that allows you to save the information. So this is the end of the tutorial. If you have any questions about how to use this finder or even about clinical trials, please contact us at this number or at this email address. Thank you.
How was that tutorial? Did people feel that they could follow this? Do we want to run a quick test case?
I think it was pretty clear.
Okay. Well, again, if we want to go to this site, it's pretty easy to go to, and you can do it in real time, and we can put, and most people do run that. Most people with recurrent disease are the ones that look for clinical trials. I would encourage everybody to look at clinical trials out of the gate. If you go to the NCCN guidelines, they also encourage clinical trials at all stages of disease. I wish there had been clinical trials when I was diagnosed. There's also this pop-up that comes up right here that you can call. And it's very easy, as Dev walked through. And I wanted to reiterate on this issue about the genetic testing here, because here's a drop-down menu. All of these alterations correspond to what you might find in a Foundation 1 report, for example. And each of these potentially corresponds to a drug that's either approved or in clinical trials. So that's why we are big proponents of getting your tumor tested. Okay.
Laura, I'm not sure if this is what you were going to discuss, but I do think that the website was really pretty easy to navigate, it seems. Would you suggest, once you see the list of these trials and the ones that are more advanced, if they're, what would be the next step to go to your oncologist and say, hey, this is what I found? I'm not sure if that's what you're planning on discussing.
Yes,
but maybe that would be helpful.
thanks, Jillian, for bringing that up, because one thing that I want to mention is that we can help you walk through, we can help you do this clinical trial finder, and then we can help you further prioritize the clinical trials and talk to you about the drugs that are in each of the trials, such that you're not taking a long list to your oncologist, but a shorter list. And also, oftentimes our doctors, they don't want to lose us as patients, right? They want to continue to help us. And sometimes these clinical trials are at a different location. So we can also help you think through like how to talk to your oncologist about why you want to be in a clinical trial and why it might be important to you. So just, I just want to give one last slide before we take additional questions. Just wanted to give this recap here, which is that most of us have been through standard of care treatments. All of us probably for newly diagnosed, and some of us for recurrent ovarian cancer. So look, I know personally what it's like to go through chemotherapy, and I know it's not easy. And, you know, I really hope that, you know, for every woman with ovarian cancer, we can each get the right treatment so we can get on with our lives. And sometimes this can involve a clinical trial. And again, I'm not a medical doctor, so I don't give medical advice, but certainly as a scientist, I think it's helpful. And, you know, running clinical trials as a profession for the last, 30 years, I've seen the advantages that it brings. And the clinical trials is how we got from Herceptin back in 1998 to all of those drugs that I showed you in the earlier slide. It's because people participated in those clinical trials. Okay, I'll stop there. Take any additional questions. Oh, I'll also just say that one of the things we didn't get into is we talked about, you know, the treatment support that we give. I didn't have time to talk about our Steps Through program, but that is free counseling with an amazing counselor who's very well versed in ovarian cancer. And here's how we can be contacted if need be, and Jillian can give my email address out as well.